Okay, good morning, everybody, and welcome to the third episode, I guess, in the our ongoing cancer symposium series, which is a joint venture between OICR and Johnson & JLABS in Toronto, Horizons and Controversies in Oncology. And I'm really excited today to chair and to get this session going on oncolytic viruses. And we're entitled this session, Going Viral, Examining Advancements in Oncolytic Viral Therapies and Their Future. And we've got a lineup of really fantastic speakers, leaders in the field, and a really exciting plenary panel discussion on where oncolytic viruses need to go and what are the gaps and what are the opportunities. I'm Laszlo Radvany, and I'm the President and Scientific Director of the Ontario Institute for Cancer Research, for those of you who don't know. Next slide, please. And first of all, I'd like to acknowledge for this meeting the land we are meeting on, and that is the traditional territory of many nations, including the Mississaugas of the Credit, the Anishinaabe, the Chippewa, and the Haudenosaunee, and the Wendat peoples, and is now home to many diverse First Nations, Inuit, and Métis peoples. So we acknowledge that Toronto is covered by Treaty 13 with the Mississaugas of the Credit. Since this is a virtual meeting, many others may be on different territories, so please take the time to reflect on and honor the Indigenous communities of your territories. Next slide, please. So this is as just a brief introduction for those of you who don't know OICR. We are a collaborative, large Cancer Research Institute in Ontario, funded by the Ontario government, 300 people strong. We're located in the Mars Discovery District. And our purpose is to really facilitate team-based research, build research networks, bring together large teams of people across Ontario to solve major problems in oncology in a collaborative mode. And our funding programs are all reflecting this. We fund both drug discovery, clinical trials, as well as research in genomics and proteomics as well, and biomarker development. And our key goal is to translate research discoveries from the bench into the clinic for the betterment of the Ontario health of its people, and of course the economic growth of Ontario. And patients and patient therapeutics and getting things into patients is central to what we do. Next slide, please. We also are uniquely positioned in this sort of mode between industry and academia, as shown here. We have a subsidiary called Fight Against Cancer Innovation Trust, which is a commercialization entity which funds small biotech startups and IP development in the cancer space to take the best discoveries and try to commercialize them and bring them, into new, cutting edge biotechs, within Ontario. And Facet has launched some of the most successful biotech companies, such as Turnstone Biologics and others that you may have heard of. So we're very uniquely positioned in that we're an institute that sort of has this academic but also industry focus to bring things into the commercial realm as well through Facet. Next slide, please. We're embarking on our next strategic plan, which is to advance early cancer research and for cancer detection and intervention research, to reinforce our global leadership in data sharing and analytics, which we've established through many internationally recognized consortia such as the ICGC. And we're going to be expanding our robust pipeline of Ontario cancer therapeutics through our drug discovery programs, both internal and our externally funded programs, and also through Facet, drive the commercialization of cancer innovations. And of course, strengthening the Ontario capacity for translational research. We spend a lot of money on IA awards and to support young investigators as well as established investigators here in Ontario, and to recruit the best cancer researchers into Ontario. Next slide. So today's topic is on oncolytic viruses, and although you'll probably get a lot of scientific introduction to what they are as part of the speaker's talks, I just wanted to give you a little bit of an introduction. Many of you know the cancer immunity cycle. in which, we want cancer cells to be killed and recognized by the immune system through the activation of adaptive immunity, and these adaptive immune cells to infiltrate into tumors. And really, we've heard a lot about this cancer immunity cycle, but I think what we're seeing now is that there's an oncolytic virus cycle too as well, where oncolytic viruses were first deployed in the clinic as just killers of tumors. People thought that we're just going to inject them intratumorally, or in some cases systemically go to the tumor, and then they're just going to kill the tumor. But then it was realized that by killing tumors, and by being able to encode things in like transgenes in oncolytic viruses, that they're more than just tumor killers. They actually can be deployed as tumor vaccines. And now we've reached the point where we can encode immunomodulators in these oncolytic viruses on top of antigens that can be encoded and on top of the tumor killing ability. So now you have this really interesting oncolytic virus cycle of them being killers, tumor vaccines, and also being able to be immunomodulators all at the same time. So it's really, really exciting. Next slide, please. I'd like to acknowledge, though, somebody, since we're always trying to celebrate the accomplishments and the contributions of women in science, especially, that when I looked back at the history of oncolytic viruses, I realized it's actually been around for much, much longer than we think. It's not a new a new concept. It turns out that one of the pioneers, probably one of the first pioneers to bring an oncolytic virus in the clinic, was from a little known country in the Baltics called Latvia in the 1960s. And the name of the researcher and clinician was Dr. Aina Musanis. And she's really considered a pioneer of oncolytic viruses. And I was really surprised when I looked up the history because I'd never heard of her. And she was a Latvian immunologist, and she was really the founder of practical cancer virotherapy. And she discovered that enterovirus or echovirus might be useful in the treatment of melanoma. And in fact, in Latvia, this virus called Rigvir, the therapy is called Rigvir, has been actually used for the treatment of melanoma for years. It was discontinued after the 1960s, but in 1999, it was restarted after national registration in Latvia. So I like to really acknowledge, this pioneer that, of the field that we forget. So next slide, please. And so we have a fantastic lineup of speakers today. We have Dr. Robert Coffin, who's really a pioneer in the field of oncolytic viruses. He was the discoverer, along with his colleagues, of T-VEC, which is the first FDA-approved virolytic therapy for metastatic melanoma. We'll have with us after that Carolina Ilkow from the Ottawa Hospital Research Institute, who's going to give a really interesting talk on her technology in oncolytic viruses. And then we're going to have a panel discussion chaired by Sam Warenke from the University of Guelph with Robert and Carolina. And then they will be joined by Catherine Taylor from Janssen & Janssen, and Johnson & Johnson, and Brad Thompson, who's also a pioneer and has done a lot of work in oncolytic viruses, who's now at Wyvern Pharmaceuticals. So I'd like to introduce Robert now. I'd like to introduce Dozy, who's the head of Johnson & Johnson Innovation JLABS, to say a few words. Dozy just joined as head of Johnson & Johnson JLABS Toronto. So welcome, Dozy, to Toronto, and looking forward to your introductory remarks.
Thank you, Laszlo. Very quickly, I've been in Toronto now a little over a month, and I've received a warm welcome, and I thank everyone there. I came from Janssen R&D, where I was involved in our product and research and development for pharmaceuticals, especially in oncology and in immunology. Next slide, please. Why would I just wanted to say a few words about what we're doing in Toronto, and for life science innovators in the audience, why you would like to partner with us. So very quickly, as you know, Johnson & Johnson have been around for over a century and driving innovations. We're the world's largest healthcare company, operating in 3 segments: pharmaceuticals, consumer, medical device, and we're in 175 countries. Just to give you a picture of scale, in 2020, we invested $12.2 billion in R&D. Next slide, please. At Johnson & Johnson Innovation, what we try to do is to curate and cultivate the world's most promising early-stage healthcare innovation. we are source agnostic. So we would like to collaborate with innovators and entrepreneurs from all over the world. And it was founded to bring the two sides of partnering, which is the innovator and industry, together in the most effective way. Next slide. This is the breadth of what we do. So we have early innovation partnering teams at our innovation centers that engage in discussions with innovators and collaborate. We also have our JLABS, which is in Toronto. There we bring access to state-of-the-art infrastructure and resources to and also mentorship and the breadth of our expertise to the community. We create connections and entrepreneurial programs. What you may not know, or some of you may not know, is also that Johnson & Johnson has a venture arm, which is called JJDC. It's been almost half a century now in existence, and it also partners with healthcare entrepreneurs. And finally, we have business development teams that partner with people at a later stage. Next slide. So here's where we are, and Toronto is in the red cluster there for East North America. So as you can see, we have a global network, and we're in centers like London, San Francisco, and Shanghai. So it gives us a global coverage and allows us to create this ecosystem and community where innovators can be connected to the breadth of our expertise around the world. Next slide. So how we partner is that we co-create. So we build a fit for purpose thing for the innovator. And this is a flexible and customized partnership model. We use our JLABS as an early stage incubation for startups, and we provide mentorship. And obviously, we also have strategic investment through external VCs, but also through JJDC. Next slide. And since we're talking about oncology today, this is my last slide, and I wanted to talk about the areas in oncology where we've been very active in the biology. As some of you know, we're invested in prostate cancer. We are looking for opportunities in hematologic malignancies, especially acute myeloid leukemia. We're also looking at solid tumors like lung cancer, bladder cancer, colorectal cancer, and looking for differentiated ideas around these areas. And obviously, we are teaching the immune system how to do new things with cancer as well, together with our partners. And finally, my email is on the next slide. If you would want to connect, or you can follow us on Twitter, and we will be happy to connect with you and see how we can partner together. With that, I'll give it over to Laszlo and for introduction of Dr. Coffin.
Thank you, Josie, and apologies for accidentally trying to skip you in the order of events here. Thank you for this really insightful opening remarks and about JLABS, and welcome to Toronto again, and I'm looking forward to meeting you actually in person. So without further ado, I'd like to introduce Dr. Robert Coffin, our keynote speaker. I really was impressed with Robert's work really many years ago when I worked for Sanofi Pasteur, where inadvertently or indirectly I ended up collaborating with him when he first developed the first HSV oncolytic viruses. We went in England, and he went on and of course has done a lot of work in further developing oncolytic viruses and commercializing them, and of course developing the first regulatory approved oncolytic virus therapy for a melanoma. He's the founder and president and chief research and development officer of Replimune, and previously he was founder and CTO of therapies, which is the company actually I was mentioning that I actually inadvertently was collaborating with. And it was a spin out from his research group at the University College London in 1999. therapies was acquired by Amgen in 2011, where Rob was VP Global Development until 2013. And before co-founding Replimune. Rob was awarded a PhD in virology from Imperial College London prior to his move to University College London in 1991. So I'm really happy and proud to introduce you to Rob, who'll be talking about oncolytic immunotherapy, past, present, and future. Welcome, Rob, and looking forward to your talk.
Thank you very much, Laszlo, and hopefully you can see my slides.
We can. They're in slide mode if you wanted to put them in full screen.
Unfortunately, I can't due to a Zoom issue, so sorry about that.
That's perfect.
But thank you very much for inviting me to speak today, and thank you very much, Laszlo, for a very kind introduction. So what I'm going to try and do is give you a bit of background, particularly on sort of how we got to T-VEC and some of the thought processes which led up to T-VEC, the data with T-VEC, and hopefully what T-VEC has told us. And then leading on from that, what we're now doing at our new company called Replimune, which is obviously intended to take things beyond what was possible with T-VEC, taking into account everything which we learnt before. So if I move on to great, it's working. So following from Laszlo's brief sort of history, it is worth going back to sort of primordial time to think about where we came from in this field. In actual fact, viruses have been used to treat cancer, or at least tried to be used to treat cancer, from at least the early 1900s, when it was at least noticed that various virus infections sometimes led to regression of cancer, particularly viruses such as hepatitis viruses, measles, and influenza. That was further developed more actively starting in roughly the 1950s, when viruses were deliberately used to treat patients with cancer. Particularly, there was quite a broad program with the mumps virus, where it was found that this could result in tumor regression. However, unfortunately, obviously the virus itself could be dangerous because these were natural pathogens which hadn't been in any way engineered or adapted from their natural form. So there was significant risk from the virus itself. And also, unfortunately, the effects generally were rather transient. We then come to the more modern era of developing viruses to treat cancer, where a more targeted approach was taken. And in the 1990s, people began using various different virus types, particularly based on herpes simplex virus or adenoviruses, which did have genes deleted from them, which should render them non-pathogenic, but still allow them to replicate in tumors. And at that time, in the 1990s, as Laszlo said, people were really thinking about oncolytics as direct cytotoxic agents. We weren't thinking about them at that time as a means to activate a systemic immune response against a patient's cancer, but more a means of just directly killing tumors. And in that context, some of the initial viruses actually were rather poor at doing anything other than that. So one of the earlier viruses was called Onyx 015, which was based on an adenovirus. And this is some early clinical data with that virus, showing a patient with head and neck cancer, which had two tumors next to each other. One was injected, the other wasn't. And as you can see, the injected tumor did shrink quite considerably, but the tumor immediately next door really didn't shrink at all, indicating that at that time the viruses being used not only were thought of as only direct cytotoxics, but were really only able to achieve direct cytotoxicity. So from that time, in the early 90s, the early 2000s, we really were trying to broaden the utility beyond just injected tumors, which is probably of relatively limited clinical utility. And we began to recognize that viruses really should have the ability to induce a systemic immune response, which should be clinically beneficial. They have underlying properties such as the fact that viruses are highly inflammatory. The immune system has actually evolved to react to microorganisms, including viruses, including both the innate and adaptive immune system. So they should be able to activate an immune system, at least against themselves, and hopefully also against the antigens which are released as a virus replicates in a tumor, and thereby systemically activate the immune system against a patient's cancer, particularly to the neoantigens which are released as a virus replicates and kills. Also, viruses have the ability, obviously, to be armed with additional genes, and viruses have not evolved to treat cancer. So it's not terribly surprising that one might not need to add additional activities to those viruses to enable them to optimally treat cancer. So T-VEC came around, it was initially thought of in the early 2000s, roughly 2001, 2002. And the rationale behind T-VEC was that it had been found in early clinical trials that tumor cells taken from biopsies and then engineered to express GM-CSF, a so-called GVAX approach, had shown some promising phase 2 efficacy data, enabling a vaccine to be created ex vivo, which was able to provide systemic benefit in a range of tumor types. However, the manufacturing of that vaccine was found to be impractical and it wasn't continued. So the rationale behind T-VEC followed from GVAX, and the real intention was to essentially make a GVAX type vaccine directly in the patient. So we intended to engineer a version of herpes simplex virus to express GM-CSF, inject the virus in the tumor, essentially melt the tumor with the virus, express the GM-CSF, and generate essentially a GVAX type vaccine directly in the patient, which should be hopefully more effective than the ex vivo manufacturing due to the direct killing as well, and also far more practical. So that was the thought process behind T-VEC in roughly 2001. And this is the structure of T-VEC. It's a herpes simplex virus with the genes for ICP34.5 deleted, which provides tumor cell activity. It also has another gene deleted, ICP47, which usually blocks antigen presentation in HSV-infected cells. And that also results in the increased expression of another gene called US11, which increases the replication of ICP34.5 deleted HSV in tumors. And then we inserted a gene for GM-CSF in place of ICP34.5. We also did want to have the most lytic virus we could get, and for that we chose a clinical isolate of HSV, actually taken from one of my postdocs at the time, as compared to lab strains, which we felt might be rather attenuating to the purpose. So this data just shows in a Petri dish that the strain 17 plus lab strain virus is a lot less effective than the strain JS1 virus, which came from my postdoc Jill Smith, at directly killing tumor cells in vitro. So we use that strain to construct T-VEC. This little bit of data on the next slide shows that upregulating US11, which I mentioned in animal models, increases the tumor shrinkage which one sees as compared to an ICP34.5 only deleted virus. As I said, we were trying to make a very lytic virus which had very powerful direct killing properties, as well as generating a GVAX type vaccine in the patient. So that clearly provided an improvement over that which had gone before. And then finally, this shows that if you do express GM-CSF in the injected tumors, you find that the uninjected tumors shrink more effectively than if you use a virus which doesn't contain GM-CSF. So these aren't the injected tumors, these are the uninjected tumors where the systemic benefit of expressing GM-CSF locally in the injected tumor can clearly be seen. So that's a sort of background of T-VEC and the rationale, and some preclinical evidence of the various components of beneficial. So T-VEC began clinical development in roughly 2003. And what we initially did was a standard phase 1 trial in all comers with advanced tumors with 36 patients, where we determined the safety profile, optimized the dosing regimen, and observed that we did indeed see anti-tumor evidence of activity, particularly necrosis and shrinkage in some of these advanced phase 1 patients. So that's shown on for an example on the bottom left, where a patient with breast cancer, local recurrence, had one of the recurrent nodules injected, and they all became necrotic and ultimately did not regrow. We then, leading on from that, conducted a phase 1B study in head and neck cancer with T-VEC combined with chemoradiation in 17 patients, where we observed a very high rate of complete pathologic response, well tolerated with durable clinical benefit. Examples of that are shown in the middle, in particular the duration of benefit seen. And we also conducted a phase 1B trial in pancreatic cancer in 17 patients, where we did see some evidence of activity, as well as evidence on the right-hand side, where tumors in the pancreas and some distant tumors shrunk in response to T-VEC. So that was the early phase 1 program with T-VEC, which then led to phase 2 studies in melanoma in particular, this line here. So we did a phase 2 trial in 50 patients with melanoma, which resulted in a 26% response rate with very good durability. And one has to remember at that time there were no approved therapies for melanoma. Things have substantially changed since, but at that time only IL-2 was available, which is, well, it isn't very broadly used, and that led to a randomized controlled trial in the same population in over 400 patients, which I'll come back to in a minute, during which time the company was acquired by Amgen. The pancreatic cancer study, while we did see some evidence of activity, we also concluded that pancreatic cancer was rather challenging, and we did not further develop in pancreatic cancer. In head and neck cancer, we were very pleased with the data we had seen, and we actually started a phase 3 trial in head and neck cancer in combination with chemoradiation. However, unfortunately, Amgen chose not to carry that on after acquiring the company in 2011, at a time when we did only enroll a handful of patients. So if we now begin to talk some of the more later stage data with melanoma, with T-VEC. So these are the results of the phase 3 trial with T-VEC in melanoma, which was an over 400 patient trial, randomized 2-to-1 T-VEC against GM-CSF. And as I said, there was no standard of care at that time, and GM-CSF was felt to be an appropriate control arm. It enrolled patients all the way through from stage 3B to 4M1C. The primary endpoint was durable response rate, which met its primary endpoint at a good level of statistical significance, as you can see, with durable responses being achieved in roughly 16, actually went up to about 20% in the final analysis of the data, and responses per se being achieved in roughly 26%, which went up to 30% in the final analysis, as compared to GM-CSF, which had a much lower response rate. So the primary endpoint and objectives of the trial were met. However, in the overall population, unfortunately, while in the final analysis overall survival was just statistically significant with a benefit of roughly 4 months, it was still a relatively modest overall survival in the overall population. However, what was found and noted was that in earlier patients with single agent T-VEC, efficacy was much greater. Patients without visceral disease had a response rate in the 40s, and patients who were first line also had a response rate close to 40. And that also translated into a much greater survival benefit in earlier patients. So in earlier patients, which is really where T-VEC as a single agent probably should be used, there's a very substantial survival benefit with a hazard ratio in the 0.5 range. So that ultimately led to the approval of T-VEC by the FDA in 2015. But as the entire population had been filed with the FDA for approval, the label did indicate that it hadn't formally been demonstrated to improve survival, even though in the earlier patients where, in my view, it should have been filed for approval, there's a very substantial survival benefit. So that's single agent T-VEC, which obviously was approved in 2015 for melanoma. But a lot of interest has resulted there is a lot of interest, particularly in the combination of oncolytic immunotherapies with immune checkpoint blockade. And there is a very solid logic and rationale as to why that should be a good thing to do, because the virus can get in and kill the tumor and release tumor antigens and hopefully create an immune response against the patient's tumor, which may not otherwise be there. And obviously, without a pre-existing immune response, checkpoint blockade can't do anything. And if the virus can really provide that may make an oncolytic virus the optimal combination partner for immune checkpoint blockade to broaden the utility beyond traditionally immune responsive tumor types. So based on that, T-VEC was entered into clinical trials with both ipilimumab and pembrolizumab. So this is randomized controlled data with T-VEC and IPI, which showed that if you added T-VEC to IPI, you roughly doubled the response rate that you which you saw with IPI alone, including in visceral tumors. And that ultimately led to the presentation of the PFS and OS data at ESMO in 2019, where you can see a modest improvement, in PFS, and also a modest improvement in survival as well. And as you can see, that really is rather modest and probably wasn't as good as one might have hoped from the very positive response rate data. But if one actually then looks at the data in more detail, you find that the patients who were on the IPI only arm went on to subsequent therapies very much more frequently and much earlier. So 64% of patients went on to a subsequent therapy in the single agent arm and only 45% in the combo, and they went on very much earlier. And one also finds that the therapies they went on to were particularly anti-PD-1 and indeed crossover to T-VEC. So in that context, where patients went on to an additional checkpoint or indeed crossed over to T-VEC, it's not terribly surprising that the survival signal was substantially diluted out. If one moves to combination with anti-PD-1, a phase 1b trial showed very promising initial data, a small study showing a 66% response rate for melanoma patients treated with the combination, irrespective of baseline CD8 status, which led to a phase 3 trial, the results of which have recently been presented, which unfortunately did show that the trial did not meet its primary endpoint as shown here. So while there was a modest improvement in PFS and OS, the benefit was insufficient to meet statistical significance and allow the primary endpoint of the trial to be met. However, it is notable that in some particular subset that there was a much greater benefit, particularly patients treated in the USA as compared to other countries actually did show statistically significant improvement in the primary endpoint PFS for reasons which are not yet clear. So questions which are still not known in relation to this trial are the degree of crossover to commercial T-VEC which occurred. And as I said, in the IPI trial, that happened in a substantial number of patients. And likewise, we don't yet know whether there's a difference in the time to next therapy, or in particular, whether more patients in the T-VEC, in the Pembro alone arm went on to IPI and earlier, as was also found in the IPI trial, more patients going on to Pembro. So we'll hopefully find out about that soon. And I personally predict there will be a lot more patients going on to IPI in the control arm than the combo arm. It's also notable that the real sweet spot patients for T-VEC, where T-VEC really works the best, which is patients with skin-only disease or in-transit disease, or that type of disease with only small visceral metastases, were actually excluded from the trial for reasons which aren't clear, but that's the place where it really works the best. So that's the phase 3 trial with pembrolizumab, which was obviously a rather a disappointment to the field in general. But I do think, as indicated, there are probably issues with the trial design, etc., which may explain that, including subsequent therapy, etc. But if we move back earlier to the neoadjuvant space, a randomized control trial has been conducted with T-VEC as neoadjuvant therapy for earlier stage melanoma. And that has shown actually a substantial advantage both in recurrence-free survival and also overall survival with a hazard ratio of roughly 0.5. So in the earlier setting, again, single agent T-VEC can be very effective, again, further demonstrated in a randomized controlled trial. So to finish talking about T-VEC, the conclusions really are that single agent T-VEC is well tolerated. It is clinically effective as a single agent, but only in patients without visceral disease, where clinically meaningful objective response rates, duration of response, and OS benefit can be shown. It also shows an OS benefit for neoadjuvant use. However, single agent T-VEC is not clinically effective as a single agent for patients with visceral disease, where response rates are rather modest, and there is no OS benefit at all. T-VEC can be safely combined with immune checkpoint blockade with evidence of clinical benefit. However, as yet, randomized controlled trials in combination with checkpoint blockade haven't demonstrated a compelling OS benefit. For IPI, that clearly was hampered by next therapies and crossover. And for combination with Pembro, the data is still awaited to see if that could have been the problem there. So the areas for improvement following all that work with T-VEC over really quite a number of years is that we probably do need a virus which builds on T-VEC to do each of the things which T-VEC was designed to do, but do it better. Particularly, we need to increase the direct tumor killing ability of the virus, increase the immunogenicity of that killing, and increase the systemic immune activation to better benefit both uninjected disease and visceral disease. And that's where we really come to Replimune, which is a company I'm now working at. And that company was set up in 2015 to really try and do all of that. So at Replimune, our platform is again based on herpes simplex virus, and we extended the hypothesis that clinical isolates of HSV may be more potent than lab strains by initially screening clinical strains from 200 volunteers who get cold sores, comparing those across a panel of human tumor cell types and identifying the one which was the most potent. And there was a huge difference between them. So we think we now have very good foundations upon which to build. And then arming the viruses with additional transgenes to really increase killing, increase immunogenicity of killing, and increase immune activation. So in our range of viral products we currently have in development, all of these viruses are based on the new clinical strain of HSV. They're all armed with a fusogenic protein from another virus, gibbonate leukemia virus, which very considerably increases killing, but also increases the immunogenicity of killing. And then our viruses RP1, RP2, and RP3 are armed with either GM-CSF in RP1, GM-CSF plus anti-CTLA-4 in RP2, or anti-CTLA-4, CD40 ligand, and 4-1BB ligand in RP3. And those are aimed to result in activating each part of the immune system or each stage of immune activation to provide the most potent systemic immune stimulation possible. So that's signal 1, signal 2, and signal 3 by each of those different products. So if we quickly move on to some data, and actually I've got far too many slides for the purpose. So this data does indicate that GALV massively increases immunogenic cell death in vitro, which translates into greatly increased activity in vivo models. So here we have 2 rodent models, a mouse model showing massively increased benefit with GALV as compared to without GALV, the virus 19 and virus 16, which translates in the ability to have very potent systemic effects in a rat model with large tumors where both injected and uninjected tumors are treated really rather effectively. If one then further loads these viruses up with the immune stimulators indicated, anti-CTLA-4, CD40 ligand, and 4-1BB ligand, one sees that the uninjected tumors in particular are benefited through the local expression of those proteins in the tumor, in the injected tumor as it dies. So this is RP2, which expresses anti-CTLA-4, and this is RP3, which expresses all of the genes together. And the panels at the bottom are to show that you get really robust expression of the transgenes in these mouse tumors, but you also get very, very, very robust killing. The whole center of the tumor has been killed off within 3 days with a low volume of virus. So this really should be a very potent systemic immune activator. So with all of those, we're in the process of developing in a number of tumor types. RP1 is in particular in broad clinical development in a range of skin cancers. But with RP2 and RP3, we're really trying to broaden out well beyond skin cancers to really hopefully deliver the promise of oncolytic immunotherapy across oncology. So just some clinical data with RP1. So our lead indication is in cutaneous squamous cell carcinoma, where with nivolumab, we've shown very deep and durable responses in CSCC, with a high response rate of 60%, a high complete response rate of nearly 50%, and activity in other non-melanoma skin cancers as well. And these responses really are truly systemic. So this is a patient with extensive disease in the neck, in the abdomen, and bone metastases, all of which have resolved and resulting in a durable complete response, with very promising biomarker data as well, showing very robust CD8 and PD-L1 upregulation, which we see routinely in our patients. This is another CSCC patient with really rather nasty disease, which, as you can see, completely resolved into a complete response. Our second lead indication for RP1, where, as I said, the focus still is on skin cancers, is in melanoma, and particularly anti-PD-1 failed melanoma. And in anti-PD-1 failed melanoma, in combination with Nivo, we've seen we're currently in the 30s response rate wise, again with very good durability of response and deepening responses over time. So there we also have a pivotal trial ongoing. And again, a couple of examples in melanoma. Again, truly systemic responses here. We're injecting deep tumors as well as superficial tumors. And the top panel in an ipi-nivo failed melanoma patient shows that not only does the injected tumor in red substantially shrink, but also all the uninjected tumors circled in yellow also do, resulting in a very durable effect. RP2 is in phase 1 and has shown a very impressive monotherapy activity, with 3 of the 9 patients enrolled showing durable RECIST responses following a short course of RP2 monotherapy, which does encourage us to believe that we can move out of skin cancer with RP2 and RP3. Again, just a couple of patient examples to make the point. A clear, nice, complete response there, and very impressive response in liver metastases of uveal melanoma in this patient. Combination, likewise, with RP2 has been promising so far in an advanced phase 1 population. We have 23 or so % response rate, but particularly in PD-1 failed disease of melanoma, uveal melanoma, and head and neck cancer, good evidence of activity with good durability. A couple of further examples of patients here. Activity does seem to be entirely independent of baseline PD-L1 status. So again, further indicating that turning cold tumors hot is being achieved with RP2. And the intention with RP2 and 3 is to move well beyond skin cancers, as I said, following the phase 1 program on the left-hand side into a phase 2 program into a range of tumor types, with particular focus on liver mets from those tumor types, which will hopefully then lead to pivotal development. So that phase 2 program will kick off roughly in the middle of next year. So to do all that, manufacturing is extremely important. So we have ourselves invested substantially in manufacturing and have a state-of-the-art facility just outside Boston, which is sufficient to cover all of our needs, including commercial needs, going forward. I realize I did put too many slides in, and therefore I'm sort of rushing. So the summary and conclusions from all of that are that, following in the footsteps from T-VEC and other earlier oncolytic viruses, we've tried to develop an enhanced potency platform, which particularly takes advantage of the ability to arm a large virus such as HSV with multiple transgenes, which are intended to augment what the virus has naturally evolved to do, which is not to kill cancer. So we've augmented its ability to kill, augmented its ability to induce immunogenic cell death, and augmented its ability to induce a systemic immune response, particularly focusing on pathways where intratumoral delivery may be most optimal, particularly with anti-CTLA-4, CD40, and 4-1BB, which is intended to get each of the stages of immune activation optimally going. So we're in registrational development when we're looking forward to the results of those pivotal activities in the next couple of years, with RP2 and RP3 following along behind, with the overall objective of expanding the utility of oncolytic immunotherapy beyond what was possible with T-VEC in melanoma, to hopefully be able to broadly address cancer across its various types, including many of the more prevalent types, which are really where we need the improved therapies to go. So with that, I will finish, and would certainly like to acknowledge the many people who are involved in this over the last 20 years, which are vastly too numerous to try and put together a slide to cover, and also obviously the patients and their families who very much contributed to all these overall efforts. So I'll finish there.
Thank you, Robert. This was a really exciting talk and really well laid out, and sort of a good overview of the field and where you're going and the power of oncolytic viruses to be immunomodulators. I think this is really, I'm looking forward to seeing more results from your clinical trials. In the interest of time, I'll move on, but we will have time for audience questions in our plenary panel discussion. I want to remind everybody that there's a tab on the bottom of your screen where there's a Q&A icon and then a raise hand icon. So if you want to post some questions that can be addressed in the panel discussion later on, please post a question in the Q&A, and then we'll make sure that we'll look at it and then we'll address it to the panel members. So I'd like to introduce you to Dr. Carolina Ilkow from the Ottawa Hospital Research Institute. I met Carolina 3-4 years ago. Really impressed by her work and her scientific abilities and know-how. She obtained her PhD at the University of Alberta. She went on to do postdoctoral work in the lab of John Bell. And then, of course, she really rose in notoriety for her really great scientific abilities and research capabilities. And she was recruited as a scientist at the Ottawa Health Research Institute. And she's an assistant professor in the Department of Biochemistry, Microbiology, and Immunology at the University of Ottawa. And she's going to talk about oncolytic viruses, challenges, and future perspectives. So take it away, Carolina. Welcome.
Thank you. Thank you so much for this invitation. I'm super excited to share a little of what we are doing, but also discuss the challenges and where I think at least my group is going using oncolytic viruses. It was a great talk by Dr. Coffin, so I will try to follow with the standards. So today I will talk about the challenges and the future of perspectives of oncolytic viruses. And I think the first thing that we need to think is, what are those challenges, and what are these considerations that we need to have to really develop the next generation of oncolytic viruses. So the first thing is, Dr. Coffin really talk about HSV and a DNA virus, but in reality is there are many different viruses out there. So one of the considerations that when we want to develop these technologies is to select the virus type. So there are viruses, RNA viruses, DNA viruses. The viruses have different complexity. The tropisms and the genomes of those viruses are very different. So one of the key considerations in the field should be, what is the right virus, and maybe what is the right virus for the tumor types that we want to target, as well as the right virus for the further engineering that we want to do in those platforms. The other, I think, key aspect in the field that we always discuss is how we deliver these viruses. I think the other consideration is not all the viruses can be delivered in the same way. Some could be injected intratumorally, some can be given intravenously, others can be a local region therapy, so they can be delivered, for example, intraperitoneally for ovarian cancers, for example. So I think the delivery aspect is extremely important. And also this aspect here, let's see if I can put my pointer. The other aspect that I think is important is, can we combine them? So there are studies that demonstrate that indeed we can use these heterologous prime boost vaccination approaches with oncolytic viruses that allowed us to combine different platforms with different features. And I think the other aspect that we encounter challenges is how we clinically test these viruses. So what is the optimal dosing? How many doses we can give to patients? Can we combine with surgery? One, we give it, what is the right regimen in those circumstances? As well as tumors are all different, and oncolytic viruses are all different too. So should we start thinking about the right platform for the right tumor type, and start considering aspects of the histology of the tumor as well as the nature of the virus to make those clinical decisions. And I think the other aspect is we know very little about not only the translation, but also the biomarker aspect. So we don't know yet what make a tumor really a right home for an oncolytic virus. So the field of discovering new biomarkers and aspects that will allow us to predict at some level what oncolytic virus will work well in certain tumor or not, I think is still an open field for us to really tackle. So these are challenges and considerations that I think many different groups around the world are thinking about it. But I think the field is doing extremely well at coming with ideas to overcome the challenges and barriers to response. And this is just a schematic that really summarizes what many different groups are thinking ahead to overcome the limitations. One is how we can overcome vector neutralization. So many viruses are, we have immunity against them. So they will be neutralized when they are given to patients. Or in other cases, we cannot do multi-dosing because of these neutralizing antibodies. So many groups around the world are thinking how we can overcome that. And in certain cases, groups are thinking about antibody-blinded viruses or pseudotyping the virus with different glycoproteins to make it different so the immune system cannot recognize and destroy these virus particles. Other groups are thinking about using cells as carriers, and I will go even beyond that. Now we know that certain viruses can be hidden in extracellular vesicles, so can we deliver them in a hiding state in cells or in EVs? Also, we can deliver intratumorally the infectious genetic material instead of the virions to overcome this neutralization. And of course, combining with antibodies or complement inhibitors that will allow the virus to indeed avoid neutralization and therefore reach the tumor and do the job. But also we are thinking and how we can boost that immune capacity that these viruses have, and people are combining doing vaccination approaches in a homologous way or in a heterologous way. So we are combining virus platforms and seeing better immune responses against the tumor because we are multi-dosing. And also we are enhancing the uptake. So the other aspect is not only to deliver the virus, but we really want the virus to go to the tumor and be uptaken there. So there are platforms, as I mentioned, that are using cell carriers to deliver the virus, special delivery devices, for example, inhalers can be used to deliver virus to the tumors that are in the respiratory tract. Using nanoparticles or extracellular vesicles for delivery of these viruses could also enhance the delivery. And finally, the other aspect is not only the delivery, but once the tumor is colonized by the virus, we really want that the virus spread there to boost the immune system, to boost the killing, but also it could be boosting the expression of transgenes, as I will discuss a little later. So my aspect of the talk was to talk about the vision and the future. And of course, different groups have different, are tackling different aspects, as I showed in the previous slide. But I will tell you what we are thinking and what we are doing in the next couple of slides. So as Dr. Coffin mentioned, many even clinical studies and groups are really exploring the use of oncolytic virus in combination. So the use of a single modified virus as a monotherapy without any payloads have not shown drastic and impactful changes in cancer treatment. So many groups have explored combination therapies. And if you ask me what, in my personal opinion, is the future, I think this is the future of the oncolytic virus field, is learning how to best combine in cis or in trans this virus with other therapeutics or with different payloads that will make these viruses really potent. So as Dr. Coffin educated us, most of the clinical trials, so preclinical and clinical studies that explore combination of oncolytic viruses have been in combining viruses with immune checkpoint inhibitors, particularly anti-PD-1 and anti-PD-L1, and have shown promising data that really demonstrate that oncolytic viruses can boost this immune activation and really immune responses when they are combined with immune checkpoint inhibitors. Now, these combinations are in trans, so this requires 2 different therapeutics that are administered to the patients, and we need to learn the timing and how these things need to be combined. Of course, combining therapies in this fashion also could cause, even though it's promising, could cause a financial burden because we are talking about 2 costly therapeutics that are being combined. So what my group and we are exploring is, can we use the virus as a monotherapy to really arm the virus to express therapeutic payloads? So in other words, when we used to think about oncolytic viruses, we used to modify them to make them tumor specific. And we used to do that by removing transgenes, by attenuating the virus, by enhancing tumor selectivity. But reality is these viruses could be also used as gene therapy vectors. So these viruses can indeed accommodate transgenes that have therapeutic capacity. And Dr. Coffin demonstrated us that it could be by the expression or insertion of immune modulatory genes, but also we could express many other therapeutic payloads that are carried by the virus and expressed in the tumor. So what we are thinking and what we're developing, and many groups are doing that, indeed the virus could act as a gene therapy vector that not only homes in the tumor, stimulate the immune system, but also could express many different therapeutic payloads within the tumor site. And those could be enzymes, could be different antibodies, including immune checkpoint antibody inhibitors, could be T-cell engagers, could be different cytokines, or other therapeutic payloads of interest. So I put this cartoon to really illustrate what I think is the capacity of OVs in this aspect. So we could think about oncolytic viruses as a workforce. So viruses can be equipped with information, and in this case, I will say therapeutic information. And we could think about the tumor now as a factory. So this factory is not only colonized by the oncolytic virus, but also the tumor is used as a factory of a second therapeutic. And in this case will be these therapeutic payloads that are expressed in situ in the tumor microenvironment and by those infected cells, infected by the OV. And this is fantastic because not only allows the local delivery, production and delivery of these therapeutic cargoes in the tumor microenvironment, but also we can really come up with creative ideas of how we can systemically deliver also these OV express therapeutic payloads. So in other words, I think the virus is able to hijack the tumor and make now the tumor a factory of cancer therapeutics. And I will give you two short examples. One is we are developing what we call VEVERs, so a very Canadian name. So these are oncolytic viruses, vaccinia viruses, that we have trained or created to express bispecific T-cell engagers. So in this case, we could create viruses that in situ in the tumor microenvironment can express these bispecific T-cell engagers, and in that case, further boost the immune response against cancer cells. And I will give you just one example of a work that one of the postdocs is doing here, but we are developing VEVERs or oncolytic viruses expressing bispecific T-cell engagers, mostly for tumors that express CEAA. And bottom line is we are able to not only express these bispecific T-cell antibodies, which we could purify from infected cells and really prove that they are functional, but also we are seeing drastic improvement in tumor control when we compare to control virus or virus that do not express this therapeutic payload versus viruses that do express bispecific T-cell engagers. In this particular case, bringing T-cells and cancer cells that express CEA on the surface and boost the killing of those cells. But the other aspect that we could think about it is, can we go even beyond a single therapeutic payload? And this is a work that another postdoc in the lab, Dr. Nicholas Martin, is doing, is can we use the virus to really express and encode a complex therapeutic payload? And in this case, we are developing technologies that allow us not only to reprogram the tumor microenvironment, but in this case, we are expressing therapeutics that have the capacity to self-amplify in the tumor. And we are doing this by expressing what we call self-amplifying RNAs from vaccinia viruses. So in a nutshell, what we are doing is we are using vaccinia because it has a lot of a great capacity to express different transgenes. And we are reprogramming the vesicles, and in this case, produced by infected cells. And in this case, we are using virus-like particles. So we have a vaccinia virus that is able to produce vesicles or virus-like particles that will contain and distribute in the tumor microenvironment this self-amplifying RNA. And the beauty of that is that from one copy, we will get many copies of RNA, and therefore many copies of the therapeutic payload. So you can imagine the capacity of boosting the expression of these payloads within the tumor microenvironment, and what we could start creating and thinking in the future for really expressing high levels or using the tumor to produce high levels of these therapeutic payloads. So this is just a proof of concept that Nicholas got, but you could appreciate in this case is a vaccinia virus that express these virus-like particles that are sent out from the infected cell and contain a self-amplifying RNA for mCherry. So this is a proof of concept. So the self-amplifying RNA will express mCherry, which is a red protein, and you can appreciate it outside the infected site here, which is shown in yellow. So we have infection, and then we have this spread, like a wave of VLPs containing this self-amplifying RNA that goes from the infected site. And this is also shown in 3D, where we have a vaccinia virus, in this case is expressing the yellow fluorescent protein, but in addition is reprogramming these VLPs that go outside the infected cell and carry the self-amplifying for mCherry. And you could appreciate that now we can reach areas beyond the infected site and at very high levels. So with this, I I just give you a glimpse of what we are doing in the lab and where is our vision. And our vision is indeed engineering viruses to really express at high level therapeutic payloads within the tumor microenvironment. But in conclusion, I will say that oncolytic viruses as a monotherapy without expressing any transgenes have shown limited clinical benefit. Many groups around the world are working to overcome these challenges with different creative tools and approaches. There are still barriers to overcome, including the neutralizing antibodies, how we can multi-dose these viruses, how we deal with hard to penetrate tumors. The combination therapies is the way to go, and many people are combining oncolytic viruses in trans, so with other therapeutic platforms, and others are genetically engineering these viruses to express therapeutic payloads, as my group and many others are doing. And finally, I will say that that safety is still extremely important. So when we do all these manipulations, we always need to take into consideration how to safely create these new viral platforms. With that, I would like to thank you, all of you, for being there, for listening, as well as all the funding organizations that support our work, and all the people that work in the lab, and in all the other groups that are making this field to grow and move forward. Thank you.
Thank you. Thank you, Carolina. And the next session would be a panel discussion with the speakers, the previous speakers, and also two additional panelists. I am Sam Workney from the University of Guelph. My research encompasses oncolytic virus, cancer immunotherapy, and immunogenic cell death. I will be moderating this panel. And first, before we proceed, I would like to ask the panelists, Dr. Thompson and Dr. Taylor, to introduce themselves.
I'll go first, I guess, since Catherine hasn't come on yet. I'm Brad Thompson. I'm working in infectious disease and cancer, both from an immunology perspective for the last too many years. And our current work is looking at gene therapies to enhance the ability of the immune system to deal with both viral infections and with cancer.
Hi everybody, I'm Catherine Taylor. I'm principal scientist at Janssen, which is part of J&J. Did my PhD in HSV biology at McMaster University. From there, went to Sanofi Pasteur in Toronto to work on new viral vaccines. Unfortunately, after that, had to leave Canada and ended up at a small startup called Benavir that works on oncolytic herpes simplex virus. It was a nice success story there. We ended up getting acquired by Janssen, and so now I'm the biology lead of our OV program.
Thank you so much. So we will proceed with the panel discussion. So how we would go about it is there is a Q&A panel where audience can type the questions, or we also have an option to raise your hand and ask the questions live. Since there are a couple of questions asked previously, so I would go with the first question. That goes for Dr. Coffin. The question goes like this: how long the Replimune virus remain active in patients? Do you see, also the second question, follow-up kind of, is do you see difference between the Replimune virus and T-VEC?
So to answer the first question, we know that in a reasonably large subset of patients, we see relatively high levels of virus in the blood of patients up to the next dose of RP, whichever RP they get. So they're given doses either every 2 or 3 weeks. And when we take a blood sample immediately before the next dose, in a largish minority of the patients, there's still reasonably high levels of virus circulating, which to us indicates robust replication. And it is actually worth noting in regard to the second part of the question that T-VEC showed much shorter persistence in the blood, only for 24 or 48 hours maximum. So we haven't done any head-to-head comparison of RP1 versus T-VEC or 2 or 3, but we do know that RP1, 2, and 3 are able to effectively treat preclinical models which T-VEC is not effective in. And I do think the clinical activity we're seeing is different to that which has been seen with T-VEC. So no formal confirmation that the RP viruses are more active than T-VEC, but all the data together indicates, at least to us, that they are.
Thank you. So the second question is a preset question from the panel, and it will go for Dr. Taylor. In the field of oncolytic viral therapy, there's ongoing debates. Should we administer the virus like intratumorally, systemically? And where do you see this goes in the clinic? Do you think one will take over the other, or are those both viable options to be applied in the clinic?
Yeah, I mean, it's such a great question. And it's a huge question facing the field right now. I mean, the good news is we already know, and Dr. Coffin showed nicely today, that intratumoral delivery works. It works at least some of the time. But if you talk to patients, if you talk to clinicians, definitely if you talk to pharma, what they want to see is systemic delivery. It's so much more convenient to give someone an intravenous infusion than it is to try to treat the tumors, especially if we're talking about a visceral tumor, because that really takes a specialist. It takes a lot of imaging technology. It might take surgery. It's going to limit the number of doses you can do. But that's not to say that IV is easy. I mean, Dr. Ilkow talked about this quite a lot, that neutralization is a huge problem. It's something we're working on, and there's some nice approaches to try to limit how much of an effect this has, but I don't think we've cracked it yet. And even if you get around neutralizing antibodies, you're still going to deal with just getting enough virus to the tumor if it's given systemically. It's such a huge sink doing it like that. The virus is going to end up in the wrong compartment. It's going to end up cleared. It's going to end up phagocytosed. And just trying in the clinic to actually get enough virus to the tumor has been a huge limitation. And so people who've been doing IT delivery for a long time are saying, why would I go to all that trouble? I mean, this is a lot of effort. And the whole idea with IT is if you have the right virus, it's going to produce a nice systemic immune response, and that's going to clear up the tumors. Why would I bother trying to get the virus infused? And again, that's a great idea, and it sometimes works, but the reason it doesn't always work, in my opinion, is just that immunosuppressive TME that we're seeing in tumors. And it's a big ask. Even if you get a beautiful anti-tumor response, you've got lovely T cells, when they try to infiltrate a non-infected tumor, a lot of the times they get shut down, they get exhausted, and they're not really working. So it brings us back to systemic delivery. If you can get the virus into more tumors, and they can do what they do and turn those tumors from cold to hot, it really helps then have your immune response do the rest of the work. So I think we need both. I think it's a great goal to keep working on systemic delivery, and we're making so much progress. But in the meantime, we already know that IT delivery can do a lot of good.
Thank you, Dr. Taylor. Anyone of the panelists have any opinions that they want to add or viewpoints?
Well, yes. I mean, I certainly agree with the barriers are challenging for IV administration. My personal view is they're likely insurmountable, both the immune response against the virus and the ability to get enough virus to tumor IV to really do any good. And I would also push back a little bit on the practicality of IT for administering to deeper tumors. The reality is the administration is exactly the same as taking a biopsy, but in reverse, and actually uses narrower gauge needles and taking a biopsy. So really anywhere you can take a biopsy from, you can inject, and anybody who can take a biopsy can also inject. And that doesn't need to be an interventional radiologist. It's just a standard radiologist generally. And very frequently you can just use ultrasound rather than CT guidance. So if you only do have to inject a few or one tumor a few times, in our view, that is practical as long as you're inducing a sufficiently potent systemic effect, which is obviously what we and others are trying to improve upon by developing new viruses and arming them up with very potent systemic immune stimulators. On the TME, however, the distant tumor TME, I do think the best way to try to address those real issues are with systemic approaches, i.e. antibody or small molecule approaches, which are going to modulate the TMA in a positive sense, using the virus purely as a means to induce the initial onslaught of T cells, which, if one is able to appropriately modulate the distant TME, should then be able to be effective. But I myself don't see IV as being the way to ever generate the army of T cells you need to do the job.
Thank you. So from both of the speakers, we have seen a lot of enthusiasm and also direct evidence that integrating the immune system into OV therapies is a promising approach. So the question, this question goes to Dr. Thompson to comment on what immunotherapy enhancing modifications do you expect in the next generation of oncolytic virus?
Oh, thanks very much, Sam. I mean, when you look at people who have cancer, I mean, they're the ones who have defects in their immune system. You know, and I mean, we all, depending on who you talk to about frequency differences, we all get cancer every week or every month or every 6 months. And most of us, our immune systems just deal with it. I mean, and you know, if you want to look at an incidence rate, I mean, it's 1 out of 1,000 people a year get the disease cancer, but 1,000 out of 1,000 usually have had some expression of cancer cells. So in the vast majority of cases, people's immune systems are just dealing with it. And so the question is being, what are the defects in that percentage of patients, that very small percentage of patients who actually express cancer? And that's been kind of the challenge as we've gone forward. I mean, as Robert pointed out, I mean, historically there were cases where people would come down with viral illnesses and you know, mysteriously all their tumors would disappear, but it was quite rare. But what you're dealing with is you know, patients who have a defective immune system. Yes, and a very small percentage of them had just enough help from the viral infection that their immune system could deal with it. So it's a pretty minor thing, and all of us who've run quite a few patients in clinical studies are a handful of, you know, the magic patients who, you know, they got a low dose intratumoral shot or they got a low dose IV shot, and all their tumors disappeared. You know, I mean, and in my case, it was the first 2 patients we treated. I mean, one was a head neck patient whose tumor just disappeared, and the other one was a non-AIDS Kaposi patient who was stage 4. You know, I got a really low dose, 1 shot in 1 tumor. It was in an early stage safety study. And so it's possible, it's there. So I think our goal is to identify what are the defects in the immune system that lead to those cancers. Now, I mean, Robert's group has covered off on most of them. If you look at defects in cytokines, you'll look at defects in checkpoint molecule expression, usually overexpressed, and look at those things. I think that is kind of the fertile area, and we're all really just so happy that Robert resurfaced after his BioVex experience and has got into that area and is doing a more comprehensive look at that. The one thing that is, I think, missing out of all the approaches is looking at some of the when you look at the viral defense mechanisms that the body has, there's some defects in there too that are more common in cancers. You know, like the toll-like receptors, TLR3. There's the majority of solid tumors are TLR3 minus, which is an unusual case. And when you look at HCC patients, when you get them treated and you look at their TLR3 status, the TLR3 minuses have a median survival of X. The TLR3 pluses have a median survival 17 times that. That's under standard any kind of immune therapy. So I mean, it's there, and that's the approach we're taking. I mean, we took a look at what Robert was doing and said, okay, he's covered up most of the bases. The one base that they're not working on, at least externally, that we could see was looking at the TLRs. And so we've developed some gene vectors that actually express TLR3, native TLR3 in different cell populations, and that should result in what we're doing. But those are the sorts of things we need to do. Look at what's missing and fix it. And that's where the field's going.
Thank you very much, Dr. Johnson. So the next question is from the audience, and it goes for Dr. Ilkow. And I would also modify it a bit. The question reads like this: how do you see the future of oncolytic virus decorated with tumor peptides? And this is a really important question with regard to personalized approach. Like, so do we have that sort of system where we can take patient tumor, sequence it, identify neoepitopes, and then put it into oncolytic virus? So what's your opinion on this?
Just need to unmute. No, it's a great question. And indeed, I think some of those approaches have been explored, right? Like, for example, the heterologous prime boost trial that Turnstone have done, it was expressing MAGE3 or expressing antigens for human papilloma virus induced tumors. So in some cases, specific tumor antigens have been explored as a vaccination approach. Now, I think there is two components, and one is you mentioned sequencing the tumors and identifying what are those specific tumor antigens and building a virus that is personalized to that particular patient or group of patients that will share those antigens. So I think it's feasible, but I think from the manufacturing point of view, and I don't know even the regulatory point of view, if that is something that is as feasible as it sounds, because it's very complicated. It will be, unless we build it in a way that can be used for a population of patients and not a single patient, I think that will be very costly and hard to do if we want to engineer in those antigens within the virus. Now, what I think the beauty of oncolytic viruses is that they act as cancer vaccines, and they act as cancer vaccines within the tumor microenvironment. And I think they have, we don't need to know, of course we could add antigens and express it from the virus, but the virus is an agnostic cancer vaccine that will infect the tumor and release all these different tumor antigens within the tumor and hopefully educate the immune system to recognize those tumor cells and kill them. So I think it can be done, but in a personalized way, it will be very hard to, I don't think it's feasible from a manufacturing and financial point of view.
Thank you very much. So yeah, I think Dr. Ilkow touched on the manufacturing and production of oncolytic virus. And since Dr. Coffin had a lot of experience with producing two types of oncolytic virus that have gone into the clinic, it would be nice to describe to the audience what are the major bottlenecks you have encountered or potentially people could encounter in terms of manufacturing, production, regulatory approvals when someone comes into the oncolytic virus space.
So, I mean, sorry to just go briefly back to the prior question. I really do think one of the beauties of the oncolytic approach is that it's off the shelf, and you should be able to make one product which is broadly applicable to hopefully a large number of patients across hopefully different tumor types. If one ends up making a different virus for each patient or even a small group of patients, the manufacturing complexity will be extremely significant, including working out what release tests you should be using, potency assays and all those sorts of things, et cetera, et cetera, et cetera. So while one may be able to prove proof of principle, I myself would be rather surprised if it was practical enough for widespread use. However, generally, oncolytic viruses are really building on the live infectious vaccine space. The most similar product which exists, in my view, to T-VEC or RP1, 2, or 3 are the VZV, live attenuated Shingrix vaccine, which is a live attenuated HSV or VZV. So manufacturing technology for those sorts of viruses or products is well established. And while it requires a lot of optimization and know-how in regard to your own specific product, it isn't requiring the breaking of new ground with regard to manufacturing technology or indeed regulators' comfort with what's necessary to produce material which can be used in humans. So, as I said, it's not about real bottlenecks or needing to develop new technology, at least for viral types for whom manufacturing has been done in the past. It's more optimization in creating one's own internal know-how, which enables one to do it sufficiently, efficiently, and reproducibly, such that you have a commercially viable product.
Thank you very much. Thank you, everyone for your active participation. And the audience for sending the questions. We will wrap up this session, and I thank OICR and JLABS for organizing this session, and we'll move on to the next session. And I will hand over the platform to Laszlo. Thank you very much.
Thank you, Sam. And I'd like to thank the panelists again, Brad Thompson, Robert Coffin, Carolina Ilkow, Catherine Taylor, for joining and a really good active discussion. I think we could have probably spent another 2 hours talking about oncolytic viruses and some of the issues facing manufacturing, the biological effects, and the huge opportunities. I do echo some of the comments that were made regarding sort of the more agnostic approach towards targeting tumor antigens. I think my observation in the tumor immunology field, as I've been in it for almost 30 years, is that we tend to go through a yin-yang where we become reductionist and then less reductionist and more broad, and then we become more reductionist, and then we realize that sometimes we're becoming too reductionist. And I think the beauty of oncolytic viruses and similar technologies is that, such as tumor-infiltrating lymphocytes, for example, is that they have a broad sweep of antigens that they can target and or release. And we know that the tumor evolves and is selected and immunoselected. So just by encoding and going after specific antigens all the time, we're chasing a loose goose, because the tumor will just expand clones that don't express those antigens or shut down those antigens. So going after more broadly applicable type of approaches as oncolytic viruses armed with all these immunomodulators, as Rob showed and Carolina showed, I think is a huge opportunity and very, very exciting. And I really look forward to how the field evolves over the next number of years to be really deployed, especially in earlier stage cancer in the neoadjuvant space. I think this is a huge opportunity, and one which OICR is really championing, and it's in our current strategic plan to sort of target more perioperative early cancer treatment interception with immunotherapies. And I think oncolytic viruses could be really well equipped for that. So I'd like to close the session for today. I thank everybody for joining, and for the 88 or so or more participants that held on to the final minutes. And again, thanks to the panel, thanks the speakers, thanks JLABS too for working with OICR. And I look forward to joining everybody and welcoming everyone to our next session in a few months on another exciting topic that we'll announce. So thank you very much, and have a nice day or a nice evening wherever you may be.